welcome to the first lecture of the 2024 GSA Awards Seminar Series. My name is Tintin Su, and I'm the treasurer of the Genetic Society of America. I'm here today with Dr. Deborah Andrew. In March of this year, Debbie was presented with the George W. Beadle Award at the Allied Genetics Conference 2024. This award honors individuals who have made outstanding contributions to the community of genetics researchers, have an exemplary research career, and strive to provide a voice for the community, maintaining active leadership roles. Name in honor of George W. Beadle, Nobel laureate, outstanding scientist, and respected academic administrator and public servant. Beadle also served as GSA president in 1946. Debbie is recognized for contributions to student education at Johns Hopkins, service to the Drosophila community through active leadership in Fly Board and the annual Drosophila Research Conferences, and acting a key role as Associate Director of Faculty Development at Johns Hopkins. Her previous and ongoing efforts to organize Drosophila researchers, teach and mentor scientists at all career levels, and build a diverse and inclusive scientific enterprise are exceptional. Debbie is Associate Director for Faculty Development in the Institute for Basic Biomedical Sciences at Johns Hopkins, where she holds the Bayard Halstead Professorship in Cell Biology. Her research explores the regulation of form and function in epithelial tubes of fruit flies, Drosophila, and provides insights into the mechanisms of epithelial tube morphogenesis and homeostasis in human development and disease. She has authored or co-authored dozens of peer-reviewed publications. She currently serves as on the editorial board for Developmental Dynamics, and her research has earned several grants and awards, including 2 ongoing NIH grants on which she serves as principal investigator. The Andrew Lab studies how organs are specified, form, and specialized using the Drosophila salivary gland and trachea as model systems. Having identified factors required to specify salivary gland and tracheal sulfates, Debbie's lab also has characterized many of the earliest acting tissue-specific transcription factors that maintain specialized sulfates, control morphogenesis, and activate tissue-specific gene expression patterns. Debbie will speak about her work, the work her lab has done and is currently doing during today's seminars. If you're just joining us late, before we hear Debbie, I have 2 quick housekeeping items. The first is that all GSA online events are covered by a code of conduct, and the link for that is available in the chat. The second is to ask you to put your questions for Debbie in the chat, and we'll get to as many as possible in the Q&A portion at the end of her presentation. And now I'm delighted to hand it over to Debbie.
Thank you, Tintin. It's such an honor to get an award from genetics, the Genetic Society of America. I can remember how excited I was to take my first genetics course as an undergraduate. It excited me so much that I left freshwater ecology, which was my major, and did a master's degree with David Kuhn, who was the instructor for that, or the teacher for that course. It's one of the best decisions I've made in my life, at least professionally. And I am so glad that he was there to offer that course in genetics. So today, what I'd like to talk about is my favorite organ in my favorite model system. And I will cover how this organ is specified and then acquires its ability to carry out its physiological functions. So today I'd like to talk about specifying and specializing the Drosophila salivary gland. As I'm sure you can appreciate, all of our organs do secretion, or all of our cell types do secretion, but there are dedicated secretory organs that are present in all of us that are doing a little more than their share. So for example, the human salivary gland can secrete up to about 1 liter of saliva per day. The pancreas secretes about 2 liters of digestive enzymes per day. And the mother of all secretors, the bovine salivary gland, can secrete as much as 32 liters at peak periods of production. And I've heard estimates up to 60 liters more recently. So the salivary gland in Drosophila is a dedicated secretory organ. It secretes more than any other cell type in the Drosophila embryo and larva, so it makes an ideal system for understanding how secretory organs are specified, specialized, and make the right kinds of products. So the questions my lab has been addressing over the many years that I've had a lab is what specifies the salivary gland sulfate, what controls the specialization and physiological activities of this organ, and what controls the process by which the primordia form the mature organ. For today's purposes, what I will focus on are the first two questions: what specifies the salivary gland sulfate, and what controls its specialization as development ensues. So we know from work in my lab and that of Steve Beckendorf's lab that there are positive factors that are required to specify the salivary gland. These factors include sex chromosomes reduced, classic homeotic gene, as well as two more broadly expressed homeotic or Hox transcription factors, EXD and HGH. So these three proteins are absolutely required to form salivary glands. And we know that because when we look at embryos that are missing function for any one of these three proteins, what we see is that for any marker of the salivary gland, even very early expressed markers, we see no evidence of expression when SCR, EXD, or HGH are missing. So it turns out that EXD and HGH, as I mentioned, are very broadly expressed in the embryo, but the Hox gene, sex chromosomes reduced, is limited to just a limited anterior domain of the embryo. So this is a beautiful slide from Bill McGinnis's lab showing the expression of all of the classic Hox genes in Drosophila. And what you can see in blue here is the expression of the SCR protein throughout parasegment 2 and only in the dorsal regions of parasegment 3. So SCR is the only spatially limited component that's required for salivary glands. So we asked if SCR would be sufficient to induce salivary glands at new places. And what we found is that if we expressed SCR everywhere in the embryo by using a heat shock promoter to control its expression, we can make extra salivary glands in the embryo. So normally salivary glands form from parasegment 2. In these embryos, we're seeing expression of salivary gland markers in both parasegment 1 and parasegment 0. But what you notice is that we're not getting ectopic salivary gland formation in most more posterior segments. And that's because in addition to the positive factors that specify salivary glands, there are proteins and pathways that limit SCR's ability to activate salivary gland formation. And in particular, one of those is a gene called T-shirt. So if we do exactly the same experiment where we express SCR throughout the embryo, in an embryo that's also missing function of the T-shirt protein, what we see is that we can now induce the expression of salivary gland markers in nearly every segment of the embryo. So SCR, EXD, and HGH are positive factors specifying salivary glands, and they can do so in cells that don't also express T-shirt, abdominal B, or DPP. And what this does for us, if we look at where these factors are expressed in the embryo, is to determine exactly where salivary glands will form. So SCR is only expressed in parasegment 2 and dorsal regions of parasegment 3, but it only induces salivary gland sulfates in the ventral cells of parasegment 2 because DPP, a negative regulator, is expressed in those dorsal cells. Similarly, when we drive expression of SCR everywhere in the embryo, because of the expression of T-shirt in parasegments 3 to 13 and of abdominal A in parasegment 14, salivary glands fail to form in those segments. Now, within the SCR expressing domain of the ventral cells, the SCR expressing domain, there are other signals that specify what kinds of cells each domain will form. So for example, EGF signaling along the ventral midline is required to specify the salivary duct, whereas signaling between the duct cells and the secretory cells specifies the imaginal ring cells, which are the precursors to the adult salivary gland. So what this means is that the combination of patterning genes that are involved in specifying salivary gland sulfates determine not only where salivary glands will form and what types of cells in the salivary gland they will give rise to, but they also determine how many cells will form the primordia of the salivary gland. And that's because these cells will neither divide nor die during the entire process of morphogenesis. So for the rest of my talk, I'm going to be focusing on these purple cells here, the ones that express SCR and not any of these other factors. These will become the secretory cells, the specialized secretory cells of the salivary gland. So SCR and its partners specify salivary glands in the absence of negative factors, and they do so by activating the transcription of a limited set of transcription factors, Krebe, Forkhead, Sage, and Hookabine. These transcription factors, in turn, some of them regulate their own expression, and others regulate the expression of other early expressed transcription factors. And this is important because the genes that specify the salivary gland cell fates disappear almost as soon as the salivary glands begin to form. So these genes are involved in maintaining the salivary gland cell fate. Now, they also have other jobs that we'll be talking about today. Kreb-A increases the secretory capacity of these dedicated secretory cells. Forkhead plays a major role in the morphogenesis of this tissue, but it also works in collaboration with the Sage transcription factor to regulate what gets made by the salivary gland, the secretome, and also control salivary gland cell survival. And finally, the Hookabine gene, which is only transiently expressed in the salivary gland, is required for morphogenesis, and more recent work suggests may also be required for cell survival. So the salivary gland transcription factors that are directly activated by SCR and its cofactors both maintain and implement the salivary gland cell fate decision. So today I'm going to focus first on how we know that Kreb-A is important for increasing the secretory capacity of the salivary gland. Now, and then we'll briefly talk on how Forkhead and Sage regulate the secretome. So as you know, the components of the secretory pathway are conserved in all eukaryotes. These include the protein complexes that bind to nascent polypeptides that are destined for secretion, proteins that carry those to the ER membrane, proteins involved in the co-translational translocation of secreted and transmembrane proteins, proteins involved in forming the complexes that cleave the signal sequence, as well as proteins involved in both anterograde and retrograde trafficking between the major secretory organelles. And many years ago, a student in my lab, Elliot Abrams, decided he wanted to know about how secretion was regulated in salivary glands. So he got the genes corresponding to the Drosophila versions of all of the secretory pathway component genes, 34 in total. And he simply looked at where are those genes expressed in embryos. And as you can appreciate from these many examples here, these genes, which we refer to as SPCGs, all show expression throughout the embryo, but we also see very high levels in the salivary gland in all of these examples. So that tells us that salivary glands become professional secretory cells or dedicated secretory cells simply by transcriptionally upregulating the genes encoding the machinery of secretion. So his next question is who or what is regulating the high level expression of these genes in the salivary gland? And what he did was to get all of the transcription factor gene mutants that were expressed in the early salivary gland and simply do in situs in the mutant backgrounds with the same set of genes that he had originally identified. And what he discovered is shown here, and it's true for all the genes, although I'm only showing 4. So across here, we're just looking at what salivary glands look like in wild type, Kreb-A mutants, and Hookabine mutants. And what you can see, which was true for all of the genes, is that in Kreb-A mutants, the levels of salivary gland genes, when we compare them to wild type, go down to the levels that we often see in nearby non-salivary gland cells. And in contrast, if you look at the expression of these genes in Hookabine mutants, the level of expression per cell is approximately what we observe in wild type. So Hookabine has no role, but Kreb-A affects the expression of every secretory pathway gene that we've looked at. These panels show where Kreb-A is expressed. It's among the earliest genes to be expressed in the salivary gland, and in fact will be expressed in this tissue for as long as it's around, so throughout larval life up until metamorphosis. So the next question was whether Kreb-A mutants have phenotypes that are consistent with secretion. So this is again the salivary gland, and what we did was to look at electron micrographs of wild-type salivary glands compared to Kreb-A mutants. And in these relatively low-level micrographs, you can see the secretory cells arranged in a monolayer around the central lumen. That's true for both Krabbe mutants and wild type. But what I'd like you to note is that the volume of the lumen and the amount of secretory content in that lumen is significantly reduced in the Krabbe mutant compared to the wild type. Now, if we look at an enlarged view and focus just on the apical surface of these salivary glands, what we observe is that there are secretory vesicles that have the same electron density as the secreted luminal content. And what we see if we count and measure these luminal vesicles is that in wild type, there are far more secretory vesicles in a given section than we observe in a Krabbe mutant. Moreover, those secretory vesicles are much larger in wild type than they are in Krabbe mutants. So this indicates that Krabbe does indeed have a role in increasing the secretory capacity of the salivary gland. The next question was whether Krabbe, whether Krabbe is sufficient to activate salivary gland secretory pathway gene component gene expression. So for these experiments, what we did was to use the GAL4 system to drive expression of Krabbe in new places and simply ask if we could drive SPCG expression in new places. And shown here is a GAL4, the yeast transcription GAL4, driven by under the control of an engrailed enhancer, and engrailed is expressed in stripes. And it's driving expression of the upstream activating sequences hooked up to the Krabbe open reading frame. And what I hope you can appreciate is that when we look at Krabbe expression in wild type, which is mainly limited to the salivary glands at this stage, that if we drive expression of Krabbe in stripes, we actually see Krabbe expressed in stripes. And correspondingly, what we observe with each of these secretory pathway component genes is expression in the salivary gland, as well as in the engrailed stripes of expression. So it turns out that that Krabbe is conserved. There are 5 human orthologs to Krabbe. They're known as the KREB3 and KREB3-like proteins. So KREB3, L1, L2, L3, and L4. And so we wanted to know how conserved the function of Krabbe might be in humans. And so we did the same experiment where you use the engrailed GAL4 driver to express each of the human versions of the Krabbe protein. And what we observe is that we can easily detect expression of these genes in stripes. And if we compare the levels of expression of these genes in wild type to the levels that we see in each of these overexpression lines, that they're all capable of driving expression of the Drosophila genes in embryos. Now, the one exception here is a version of the protein where a juxta-DNA binding domain has been deleted, and this domain is conserved among all family members. And if you delete that domain, you can no longer activate expression. So what this tells us is Krabbe is not only necessary, but sufficient for driving high expression of SPCGs to increase secretory capacity, and that Krabbe regulation of SPCGs is highly conserved because we can put the human genes back into a fly embryo and they can activate expression of every SPCG we've so far tested. Our next question is whether Krabbe was dedicated to secretory function, and a question of how many of the SPCGs does Krabbe regulate. So for these experiments, we did microarrays comparing RNA from wild type embryos to those of Krabbe mutants. And shown here on this volcano plot are genes that go down significantly in a Krabbe mutant and genes that go up. And what you can see, encouragingly, is Krabbe goes down. That's good. That's a good control for this experiment. But what we also see is that all of the SPCGs that we looked at by in situ hybridization are also down in our microarray analysis. So we found a total of 383 genes that go down at least 1.5-fold in a Krabbe mutant compared to wild type. And if we do gene ontology analysis, we can determine what kinds of genes Krabbe affects. And what we found is that 116 of the 383 genes encoded known components of the secretory pathway or encoded secretory cargo. Similarly, there was a large unknown class of genes that were regulated by Krabbe or that required Krabbe for expression. And when we did a Wolf p-sort analysis to ask where those proteins localize, many of them turn out to be secreted or plasma membrane proteins, some of them localizing to the ER. Indeed, some of the ion channels and transporters that we found in this analysis also turn out to be organelle specific, so localizing to the ER and Golgi, suggesting that they also have a role in secretory function. So our next question was, how many of these genes, or does Krabbe directly regulate the secretory pathway genes? And to do this, we did in vivo DNA binding assays, so ChIP-seq, and I'm sure you know how this works, but basically you use antibodies to the transcription factors to pull down the DNA that's been sonicated to a small size, and then immunoprecipitate the antibody along with the bound DNA, release the cross-links, PCR amplify, and do next generation sequencing. And when you take the data of sequences that come down with the antibody recognizing this protein, what you can see are identify the genes that are bound by Krabbe, for example, and the sequences that are enriched in this data set. So we did this with Krabbe. We did it in a little bit of a different way. In one experiment, we used the Krabbe antibody for ChIP-seq, and that will tell us everything that Krabbe is binding to in the whole embryo. And then we also expressed GFP tag versions of Krabbe using 2 different salivary gland drivers that have in common only expression in the salivary gland, so that by looking at the shared peaks, we'd be able to identify salivary gland specific binding events, whereas the Krabbe ChIP-seq would give us binding in the whole embryo. So it turns out that Krabbe binds a very large number of sites in the genome. If we analyze these data, what we find is that Krabbe likely binds sites, different sites in different tissues represented here, and that the salivary gland binding can account for a significant portion of the Krabbe binding. So one assay of whether the experiment actually worked is to see which sequence motifs are enriched in the DNA that came down. And what we find is that they, based on in vitro DNA binding studies by 3 different labs, what's known is what Krabbe recognizes and prefers in vitro. And what we found when we looked through the salivary gland specific set of binding events is that 95% of the fragments that came down in the salivary gland contained at least one of these 5 motifs. And in fact, what we think is required for Krabbe binding is simply that there be open chromatin and the presence of at least a single Krabbe motif in that DNA. So we showed that all of the SPCGs, in fact, are bound by Krabbe, but we also identified a number of new targets, including the XBP1 gene. And this experiment is just demonstrating that the Krabbe motifs within the XBP1 gene are required for Krabbe regulation. So what we did was use CRISPR-Cas9 and homologous recombination to replace the consensus Krabbe binding sites within the control region of the XBP1 gene, and then looked at what happened to XBP1 transcripts in these situations. So it's a little hard to see here, but what you can see in wild type is enriched expression of XBP1 in the salivary gland that disappears in a Krabbe mutant. And if we drive striped expression of Krabbe, we can see striped expression of XBP1. But if, for example, we have these binding site mutants, we don't see expression in the salivary gland, and we cannot activate expression of the XBP1 transcript when we drive expression of Krabbe in embryoled stripes. So the consensus motifs are required for Krabbe regulation in vitro. We looked at a number of genes in this way, either looking at what happens. So these are genes that are all bound by Krabbe, and look to see whether Krabbe was necessary and or sufficient for their expression. And certainly the genes that were identified as bona fide SPCGs were affected by loss and or overexpression of Krabbe. But what we found is that many of the genes that are bound by Krabbe are not regulated by Krabbe. So Krabbe binds far more genes than it regulates. And so the next stage of our analysis is to try to understand what distinguishes a Krabbe bound and regulated gene from a Krabbe bound and unaffected gene. And so to begin to address this question, we've started doing single cell RNA sequencing of wild type and Krabbe mutant embryos. Encouragingly, with our wild type samples, we were able to identify all of the major cell types in the embryo, including the salivary gland. And similarly, our data isolated from Krabbe, our single-celled data from Krabbe mutants also easily identified a salivary gland population of cells. So with these data, we can now look across all of the tissues and, for example, learn what levels of Krabbe are expressed in different tissues. So for example, you can see here that Krabbe, that the salivary gland expresses the highest levels of Krabbe RNA. But similarly, we see expression in the amnioserosa, expression in the fat body, and in the plasmatocytes. Whereas tissues like the CNS and the somatic muscle express nearly undetectable levels of Krabbe. And this is consistent with what we see with antibody staining with the Krabbe protein. So we can now take those tissues and ask what classes of genes are most enriched in the different tissues. And like the salivary gland, where this SPCG genes involved in the secretory organelles, are the most highly enriched gene set, that's also true for the amnioserosa and for the plasmatocytes. Whereas if we look at other tissues like the CNS and the somatic muscle, we see that they express genes that are completely unrelated to secretion and are more in keeping with their function. So this allows us to now look to see in these different tissues, if we compare expression levels in wild type to Krabbe mutants across a large number of SPCGs, what we can see, where this dark red, this is a scale bar over here, the red is showing you genes that go down in a Krabbe mutant, whereas the light blue would be genes that go up. What you can see is that in the tissues that express the highest levels of Krabbe, the amnioserosa, fat body, plasmatocytes, and salivary glands, these are the tissues where the SPCGs go down the most, in keeping with the idea that Krabbe not only regulates SPCG expression in the salivary gland, but also in other cell types that undergo high levels of secretion. So our next goal is to take the DNA binding data that we have and compare it with the expression levels that are found in each tissue and try to crack the code of what's the difference between a Krabbe regulated and bound gene versus simply a Krabbe bound gene. So that's the data that we have supporting the role of Krabbe in activating and controlling the capacity, the secretory capacity of the salivary gland and other secretory organs. And in the final few moments, what I'd like to talk about is how forkhead and SAGE work together to regulate the secretome. So SAGE, unlike every other transcription factor that I've been talking about today, is expressed in only the salivary gland. And like Krabbe, it comes on early and it stays on as long as this organ will survive. And in fact, it's one of the most rapidly evolving of the transcription factors. And by the time you get to humans, there's at least 6 proteins that are quite similar. We knocked SAGE out by homologous recombination. And then we did microarray analyses of both SAGE null and embryos where we overexpress SAGE to find genes that depend on SAGE for their expression. And this volcano plot again shows you genes that go down in a SAGE mutant and genes that go up. You can see that SAGE is here as well as a large number of other genes. So of the genes that change the most in the salivary gland, what we find is that 36% of those encode predicted secreted proteins, 20% encode predicted transmembrane proteins, and 25% encode enzymes that are predicted to localize to secretory organelles where they modify proteins that are going through the secretory pathway. So approximately 80% of genes that are most affected by the loss of SAGE encode either proteins that are secreted or transmembrane or modified. So here is a couple examples of genes that encode secreted proteins. This one goes down 57-fold in a SAGE mutant. This one goes down 24-fold in a SAGE mutant. Both of these, again, encode secreted proteins. This is an example of a Golgi-specific enzyme. You can see high-level expression in wild type that completely disappears in a SAGE mutant, in keeping with this being a SAGE target from our studies in the microarray studies. Now, we've also looked at what happens to the expression of SAGE targets when we drive SAGE everywhere. And what we observe if we use a tubulin GAL4, a ubiquitous GAL4, to drive expression of SAGE, is that we can drive expression of these SAGE target genes, but only in a limited number of cell types. And it turns out that the cells where we can drive ectopic expression, their ectopic expression with ectopic SAGE, are cells that also express the fork head transcription factors. So shown here is the proventriculus, hindgut, and Malpighian tubules. So this suggested that perhaps fork head and SAGE are both required for the expression of SAGE targets. To address this, we looked at these genes identified based on SAGE microarray to ask if they were affected by fork head. And as you can see, the expression of all of them disappear in a fork head mutant. So I told you earlier that fork head is required to maintain SAGE expression. So the question was, is fork head working with SAGE or simply upstream of SAGE to regulate its target genes? So again, we're going to return to the experiment where we drive expression of either SAGE, fork head, or both proteins using the engrailed GAL4. So we'll be expressing those proteins in engrailed stripes. And what we find when we drive SAGE expression in engrailed stripes is we get ectopic expression of SAGE targets only in a subset of cells in the hindgut. If we drive ectopic expression of fork head, what we observe is that we get ectopic expression in cells that have been transformed from duct to secretory cells, but not much else. And it's only when we drive expression of both the SAGE protein and the fork head protein that we observe ectopic expression in all of the cells that are expressing these proteins. So what this suggests is that SAGE and fork head work together to regulate SAGE target genes. And we think this is, we've kind of confirmed this. We've done a low resolution genome-wide assay to ask whether fork head and SAGE bind to the same sites in the genome. And this is looking at salivary gland polytene chromosomes that have been stained for the endogenous fork head protein or the endogenous SAGE protein. And what you can see is that there's nearly a perfect correspondence between where fork head localizes and where SAGE localizes, suggesting that these proteins work together to regulate their downstream target genes. Now, this is in third instar larvae, and it's pretty low resolution. These bands are tens to hundreds of kb in size. And so we've gone back to do ChIP-seq with SAGE and with fork head, as well as with a shared downstream target gene known as senseless that also binds to the same sites in the salivary gland. And this is what we found. So when we do ChIP-seq with a fork head tagged with GFP, we find, again, a large number of sites that are bound, but the number one motif that comes down with our fork head ChIP-seq is a in vitro consensus binding site for fork head. If we do this experiment with SAGE, the number one site that comes down is the in vitro consensus binding site for SAGE protein, first demonstrated by Kevin White's group. If we pull down with sense, surprisingly, we pull down the fork head consensus as the number one motif that comes down. But what I'd like to point out are these other motifs that come down when we pull down on each of these proteins. So for example, the fork head consensus site comes down when we pull down as SAGE or senseless. The SAGE motif comes down with fork head, SAGE, and senseless. And the sequence that we think sense might be working through comes down with fork head, SAGE, and senseless. And what this experiment tells us is that these proteins are also working together in the embryonic salivary gland, and that they're binding within 200 to 300 base pairs of each other, because that's the average size of the DNA fragments that we created for doing the ChIP-seq. So we think fork head, SAGE, and sense all work together to control what were first identified as SAGE targets. And these SAGE targets, we believe, are primarily the secretome. So what I've told you today is about some early functioning transcription factors that work together to induce salivary gland cell fates by activating the expression of a limited number of early transcription factors. These transcription factors regulate their own expression, they regulate the expression of each other, but they also have a role in, they have specific roles in how the salivary gland develops. So I've discussed the role of Krabbe in increasing the secretory capacity of this dedicated secretory organ. And I've told you about how FORCAD works with SAGE to control the secretome. Now, there's a few lessons we can take from this. It looks like the regulatory hierarchy from specifying an organ at this layer to turning on the transcription factors that are required to build the salivary gland is not very deep. So after this level of transcription factors, we're actually seeing the effector molecules, the genes that actually increase secretory capacity or that are contributing to the secretome. So, most important slide, I would like to now acknowledge the people who've done all the work. I could not talk about other aspects of what we do in the lab. I did not talk at all about our studies on salivary gland morphogenesis, tracheal development, work on the mosquito salivary gland, and I apologize to former lab members that we couldn't get there. But I just want to say Elliot Abrams, a student in the early 1990s to early 2000s, began all of this work on Krebe, SAGE, and FORCAD. And this work continued through the decades by other lab members and is currently being worked on by Dorian Jackson, Dan Peng, and Nate Loeffner. So thank you very much for listening, and I'm happy to take questions.
Thanks very much, Debbie, for an excellent and very, very clear talk. And I learned a lot. And Debbie will be happy to take questions. So if you have any questions, please put them in the chat, or use the raise hand feature. Or
Everyone should feel free to also email me if you have any questions. Happy to.
Perhaps while we're waiting for people to type and so on, I'd like to start off with a question. Oh, wait, hang on. There's somebody has a hand raised. So, Daniel?
And
Hi, really nice talk. Enjoyed it. So I have a question about your single cell sequencing data, where you show that in tissues where there is high CREP expression, there is lowering of all the SCPGs, but there are some tissues where CREP expression is lower, but it still binds something in those tissues, but the expression of SCPGs go up. So
yes, so I would say that I think the amount of binding, so those genes are not necessarily being bound by Krebe in those tissues. I think it depends on whether the chromatin is open or not. I think Krebe is only binding when it's expressed there, right? And some tissues have nearly negligible levels. So the genes that appear to not change or go up slightly, those are probably indirect consequences of Krebe being missing in some of the tissues. So they're largely, I would say, they're not changing much, but they could be when Krebe is not there and signals are not released. There could be very indirect consequences. So I think that's my best answer.
Thank you.
Laurel?
Hi, Laurel.
There we go. Sorry, I'm slow this morning. That was a great talk. I loved the summary. I'm curious because there's a lot of localized secretion in specific tissues, but not at the level of a secretory gland. So like local secretion from imaginal discs, et cetera. So do you think Krebe is involved at some low level in regulating the secretory pathway in local, very small numbers of cells or low levels everywhere?
My bias is that there are housekeeping genes that Krebe is likely to work with to boost secretory gene expression, and that it's these housekeeping genes that are functioning in most cells, and that the amount of Krebe that individual cells make will then add work with those factors. And we're actually hoping that we can identify these housekeeping factors simply by looking at what other motifs emerge with the Krabbe bound DNA. And we're actively working on that now, trying different permutations of what regions we look at, what comes down, et cetera. So I hope to answer that better at some point, but I think the housekeeping genes do that, and then Krabbe works with the housekeeping genes would be our current favorite model. So thanks, Laura.
Debbie, there are questions in the chat. If you'd like me to read them out, let me know, or you could look at
That
them.
would be great. I will probably shut down the system if I read them
Okay.
out.
The first chat question is from Michelle Arbeitman. Are your genes that encode products that are secreted expressed elsewhere, like in reproductive tissues? What regulates secretion in those tissues?
I suspect it's Krabbe, and we haven't looked so much at, for example, the testis or the ovaries. We haven't looked in adult tissues, but Krabbe, I'd have to go back and look at that, the data on flybase, but I think it's expressed in many of those tissues. So I suspect it's doing it in many tissues, and even some of the tissues that you're not seeing expression early in the embryo, later on when those tissues begin to function, I think Krabbe is likely to be active. And I think, for example, the nervous system, not much going on in the embryo, but later on Krabbe is functioning. And I forgot the group that's been looking at that, but so I think when tissues become specialized or need to be secreting, the Krabbe is often the factor, maybe always the factor that comes up. So thanks, Michelle.
Yeah, thank you. And I just wanted to wish you many congratulations on a beautiful talk.
Thank you.
We'll take a question, the next question from chat, and then go to that raised hand. So the next question from chat is Mira Sundaram. Can you tell us more about the salivary gland secretome? Does the salivary gland have an apical extracellular matrix?
It absolutely has an apical extracellular matrix. In fact, there are proteins that get modified by the enzymes that normally modify collagens. And if those modifying enzymes are not expressed, the salivary gland will actually collapse on itself. So there is an apical matrix there. It is very important in early development in keeping the tube open. Some of the other things, the ones I showed, for example, one of them looks like a mucin, that first gene that changed by 57-fold in a SAGE mutant. That looks like a mucin. It's a very small serine threonine rich protein. Others look like they might be antimicrobial type things, acidic proteins that are made in the salivary gland. And Norbert Permon's group has shown that there's actually some basal secretion from the salivary gland. So there's a factor important for growth of the larva and adult that are expressed and require a salivary gland for secretion. So there may be, what we've mainly looked at are things that are likely to be apically secreted, but there may be things that are secreted into the animal and important for overall physiology. So,
do you think there's collagens on the apical side?
no, I do not. I think there are proteins that have the same triplet that's required for these prolyl hydroxylases to be as substrates for these. I don't think it's collagen, because the collagen genes are made in flies. They're made by the hemocytes, the plasmatocytes. And so no, not collagen per se, but proteins that have domains that look like collagen. So no collagen there, unless it's carried across the cell and deposited, but we see no evidence of that. So
Thank you.
We'll go to a raised hand in the audience. Robert Levis, would you unmute and just ask your question?
Okay, yeah, I'm old enough to remember that back in the 1980s, the Beckendorf lab studied salivary glue proteins. And I was wondering what the regulation is of their temporal expression at the time when the larvae is attaching to the substrate.
So I can't remember for sure whether they're regulated, whether it's known that they're regulated by the transcription factors we've been looking at. I suspect they are, but I also suspect that you need input from the ecdysone signaling pathway in order to also regulate those proteins. But it is the case that both forkhead and sage stay on in the salivary gland pretty late into metamorphosis, so they can still be regulating I suspect they are regulating the glue proteins that you're talking about. They don't disappear until much later relative to other tissues, the salivary gland and these genes, which in part it's because they're required to keep the salivary gland alive.
So I guess your lab has mainly studied the specification of the salivary glands.
Specification and function early, and that's kept us busy. But yes, I think it's equally important. Andy Andrus has been looking later, so you might want to look at some of his work on the late salivary gland. Thank you.
Thanks.
Going to the chat, Thomas Klein says, you've developed a remarkably complete view of this pathway. I'm wondering what aspect you found most surprising.
Tom, thank you. Thank you for listening. I read all your papers when I was just in Bruce Baker's lab doing graduate work. Anyway, what's most surprising? So I guess the parsing out of function. I didn't know that it would work out so cleanly. So when Elliot was first looking at the transcription factor mutants and beginning to see was there a pattern emerging, the fact that every secretory pathway gene he looked at had the same change in Krabbe mutants. I think that was quite surprising. Some of the other things that we're finding have to do with morphogenesis, which I find surprising that everything that we're looking at so far seems to, at least in terms of making tubes, seems to involve two major proteins and modifying those two proteins. So I think the simplicity and elegance, I think, of this system is a little bit surprising that it's not crazy. Hi, Tom.
No, I was struck by the shallowness of it. You get down to the effectors so quickly. I mean, you mentioned that, but it really is amazing.
It is, and
These are
it's true for
similar to-
morphogenesis and for, the stuff I talked about today. Yes, you don't have to go layer after layer after layer to get to the things that are doing the real jobs, right? And not just the regulators. So yes.
Very nice. It was good to hear you. Congratulations, by the way.
Thank you so much. Nice to talk with you.
Yeah. And I should add also that there's a lot of congratulatory messages in the chat, and I'm saving the chat and I'll email it to you, Debbie.
Thank you so much.
Yeah. So you get to see them all.
Awesome.
Questions more in the chat. I do have a question. It's about something on one of your slides, but you didn't talk about, and that is that under SAGE, you also have survival of the salivary gland cells, and it's not under forkhead. So a couple of questions. How does SAGE ensure that salivary gland, what's the mechanism for SAGE to salivary gland survival, and how come forkhead is not doing that with SAGE for that aspect?
Oh, I didn't. So that arrow that's going over there is to the secretome and to survival. Okay. Both forkhead and SAGE are required for survival.
Oh, okay.
Okay. And they do work together. And the way they seem to work is by keeping Reaper and HID from being expressed in the salivary gland. And the experiments that we've been doing are in rescued animals. So we rescue them from dying so that we can look at what happens to target genes. But what we see is that in a forkhead mutant, the salivary glands would normally die right at the stage where they're invaginating. In a SAGE mutant and in a sense mutant, which is also working, the salivary glands die somewhere around mid-embryogenesis, okay? And we think our hypothesis is that because when you lose forkhead, you also lose SAGE and you also lose sense because they're forkhead dependent in terms of maintaining expression, that you have a much more profound effect than when you just take away only SAGE or only sense. But the mechanism seems to be, so we can see both Reaper and HID are expressed at very high levels in the salivary gland in a forkhead mutant very early, and in a SAGE and sense mutant later. And some of this is work from Steve Beckendorf's group showing that SAGE and sense do this. So I think perhaps because this is a non-traditionally dividing cell population, that keeping the cells alive is an active process.
Mhm.
And Forkhead and its partners keep death at bay. And I think Roger Caress's lab has also shown that you can, if you knock down Forkhead at any stage during larval life by RNAi, that you can actually cause the salivary gland cell death immediately. So it's an active process keeping the salivary glands alive. And it's again by repressing the pro-apoptotic genes, Reaper and HID. So at least those are the ones we see come up. So
Can I just follow up and ask if those Forkhead and Sage bind to Reaper and HID regulatory regions, or do you think they're working through something else? So is it a direct effect or an indirect effect?
I can't answer that question right at the moment, but I can go talk to, I just don't remember whether we've looked and what we've found in terms of binding, but I don't know. I don't know the answer, but I'll email you an answer.
Okay, thank you. And we have, I'm making sure that I didn't miss something. Yes, so there is something, a question in the chat from, sorry if I mispronounced the name, Shalini Dangadurai. Wonderful work, congratulations. I'm curious about the role that crab A plays in the mosquito salivary glands. Are they required for the propagation of viruses in the salivary glands of mosquitoes? Thank you very much.
That's an excellent question, and I have no idea. What I do know is that mosquitoes have two cribs. They have one that looks a little more like the human versions, which have to be cleaved in order to go to the nucleus, which we use the cleave forms for experiments. And there's a version that looks more like the Drosophila crab A protein. So crab A is likely to be important in secretory function, but what happens beyond that, I don't know. The gene that we are targeting in the mosquito salivary gland is SAGE, and in part that's because it is the most rapidly evolving of the transcription factors. It's only expressed in the salivary glands of mosquitoes as well. So with some minor expression in the testis. We'll try to hope it's not a problem.
There are no more questions. Can we have a round of congratulations and applause for Debbie for a wonderful talk?
Thank you so much. Thank you, Tintin, for doing this, and thanks to the GSA for the recognition. It's so nice to be recognized by the people you care about the most. So thank you.